Role of virus in biotechnology. To understand the role of virus in SCID treatment. Extensive research in biotechnology involves manipulation of microorganisms for human welfare. Here, we can see a patient suffering from severe combined immunodeficiency disease, or SCID, lying in a sterile environment. A closer look at the patient's body shows that it is constituted of numerous cells, and zooming inside a single cell shows a prominent nucleus which has numerous chromosomes arranged in pairs. These are called homologous pairs. One of the genes is responsible for synthesis of an enzyme adenosine deaminase, or ADA. This gene is recessive, and its allele situated on the homologous chromosome is also recessive. Hence, the gene is homozygous recessive. Thus, the enzyme is not formed in these patients, and they face severe immune deficiency. In order to cure this disease, a virus called adenovirus is utilized. This virus has a proteinous capsule with fibers extending from its surface. On making the capsule transparent, we observe the core containing the nucleic acid DNA. This DNA is isolated from the viral core. It contains two genes. Let us mark them as A and B. Gene A encodes a protein which allows this virus to insert itself into the host's genome. Gene B causes the disease this virus is associated with. A restriction enzyme lying in the nucleus cuts gene B at 2 points and replaces it with a desired gene which encodes the formation of ADA. Let us mark it as C. So the genetic constituent of virus changes from AB to AC. This recombinant DNA is again incorporated into the viral capsule. Now the virus is injected inside the bone marrow. Here, it binds to the bone marrow stem cells through the receptors and injects its DNA. And the DNA finally moves inside the nucleus through the nuclear pores. Its DNA lies free in the nucleus along with the host DNA. These stem cells containing recombinant DNA are isolated and are cultured in the petri dish. Here, the stem cells divide, and in this process, the gene for ADA, which is present along with the viral DNA that lies freely independent of the host DNA, also divides and gets distributed. Cultured stem cells now contain active genes for ADA enzyme formation. These healthy stem cells are again injected back into the patient's bone marrow, where they start forming healthy lymphocytes. These lymphocytes divide normally in the presence of enzyme ADA, and immune system of SCID patient gets restored and the disease is cured. Recap. Let us sum up the topic quickly. When we look at the chromosome of an SCID patient and look at the chromosome, we can see the homozygous recessive alleles of a gene called ADA, which is non-functional. In order to cure this disease, adenovirus is utilized. DNA of the virus is isolated, and its disease-causing gene is replaced by a healthy gene of ADA. This virus containing the recombinant gene is made to affect the bone marrow. Here, the virus attaches to the stem cells present in the bone marrow. These stem cells are then isolated and cultured in a petri dish. Here, stem cells divide and viral DNA containing the dominant ADA genes also divides. These healthy stem cells are then injected back into the body of the SCID patient. Here, they start forming lymphocytes that could divide normally, and thus immune system of the patient gets strengthened.